off to a bad start, but thanks for inviting me. As said, I'm Eveline de Zeeuw, and I used to be assistant professor at the Department of Biological Psychology at the VU. And that's why I was invited here, because during that time I linked data from the Netherlands Twin Register to microdata from Statistics Netherlands. And we did that as a pilot to test the recently developed ODISSEI secure supercomputer platform. And that platform actually brings, I'm happy to tell you a bit about it, because it's actually providing a lot of opportunities to link different data sources to each other and allow us to answer new exciting research questions. So ODISSEI stands for Open Data Infrastructure for Social Science and Economic Innovations. And it's a collaboration between CBS, SURF, a company that provides high performance computing, and many longitudinal research cohorts in the Netherlands. But first, let me introduce you to the Netherlands Twin Register. I gathered that most of you are from FGB, so you probably already know a bit about it, but it was founded in 1987 and over 200,000 people are currently taking part. They're all a member or a multiple or a member of a multiple family. And young twins, they are we did longitudinal data collection by 8 specific surveys on the development of the children. So parents fill out surveys, and when the twins are in primary school, their teachers are also invited to participate. And then when the children turn into adolescence, they themselves can start to participate. And then we also have adult twins that participate, and they do not receive age-specific surveys, but they receive a survey on health, lifestyle, and personality every 2 to 3 years. And then in subgroups, well, we also have collected data on, for example, IQ, EEG, MRI, cardiovascular measures. And also, in addition, we have DNA and other biological samples. So here's an impression of the type of data that the NTR has on the right side. You see that's all data that has been collected directly from the participants. And on the left side, you actually see Statistics Netherlands, which has a quite different type of data. They have registered data, so they have data from many organizations, for example, on income, on migration, on educational attainment, and that they have that for almost everyone in the Netherlands. So if you would actually combine these 2 types of different data, it allows you to answer many new research questions that were not, we could not answer before when we had, when we didn't link these data together. So this, for you who are already familiar with the CBS remote access environment, this is already possible. So you can link cohort data to Statistics Netherlands data, but this environment has a limit with regard to the size of the files that can be uploaded, and with regard to how much computing power there is. So that actually prevents large-scale analyses. And for example, the NTR, I told you we have genotype data, epigenetics data, and these data are too large to be uploaded to Statistics Netherlands. In addition, cohorts can link their data to CBS currently, but they cannot link their data to CBS as well as to another cohort. So if you want to, for example, analyze the same CBS measure in both cohorts, it's not possible to do that. You have to share your data outside of the safe CBS environment, and then you would need to upload to CBS, but that's not always feasible. So the ODISSEI secure supercomputer actually solved those 2 shortcomings, but it's important to note that this OSSC platform, it's hosted on Cartesius, which is the supercomputer, and that is actually Linux based. So it's not like there's a Windows interface. So what you would, so what you have to keep in mind that, for example, you cannot use SPSS on that system. So, okay, what we did to test the OSSC platform was that we linked genotypes from the NTR to data on healthcare expenditure from CBS. And the research question that we wanted to answer was which genetic variants are related to differences in healthcare expenditure. Not because we thought we're going to find genes that explain differences in healthcare expenditure, but we wanted to use genetics as a research tool to get more insight into the association between specific traits and diseases and their link with healthcare expenditure. So for this purpose, we run a so-called genome-wide association study. I will not go into any of the methods that we used or the results of this study, but I will tell you something about how we linked our data. So NTR genotype data are actually already stored at SURF, and other cohort data are stored at the NTR, and like, for example, age and gender. And then at CBS, we had the healthcare expenditure data. So this means that we actually had 3 different locations where our data were, and all these data sets, there were identifiers in those data sets, but they were all different identifiers, and we had to link them all together on the OSSC platform, but not before all those identifiers were encrypted to ensure that in the linked data set, no one could identify which participant was which, to protect the privacy of our participants, of course. So this figure actually describes the complete procedure of linking these data sets, but I will not go into details. I'll just explain to you the most important steps that we took. So that was linking NTR cohort data to CBS data on healthcare expenditure, and then subsequently also linking the genotype data to that. So we started by taking data from the NTR participants for whom we had genotype data, and then we selected the people that had given us permission to link their own data to register data. And that was actually quite a challenge because we have been collecting data on genotypes for many years, and we had to figure out which consents were which informed consents contained permission for linking and which didn't. So we had to make sure everyone had given the correct consent. So to link those two together, we used the so-called A number, which is the number from the City Council Center. And we used that to link our participant to CBS data, because both the NTR and CBS have this identifier. And that's a unique one. So that was actually quite an easy step. If we would not have had this A number, we could have used personal data like postal code, initials, date of birth, etc. But with twins, that's always quite tricky because a lot of those are similar in the twins, especially if you talk about young twins. Even some twins even have the same initials, so then all the matching variables are the same. But that step was actually not that difficult because we had this A number, so we were actually able to link all the participants to CBS data. The next step was then to link the NTR genotype data, but there was a complicating factor because these data, they're already stored on the supercomputer, and they're stored in a binary format, and they do not contain any identifiers. So the order of the file is the implicit identifier. And when we link to CBS, the NTR identifier got encrypted and the file got reordered. So because we did not want to be able to identify people after linking with CBS, but that also meant that we could not analyze our genotype data. So in the end, we actually solved this problem by allowing SURF to take on the role of trusted third party, and they pseudo-randomized the order of the genotype data and then provided us with the key between the NTR identifiers and the new order of the genotype data. And this complete procedure ensured that none of the parties involved had access to all the linkage keys, and neither could identify participants in the cohort, nor in the administrative data, nor in the genotype data. So to verify that it actually worked, because it was a lot of steps and we had a lot of different identifiers, and we also pseudo-randomized the genotype data, what we did was that we also run our analyses on height as reported by the participants. And we had height, we uploaded that together with the NTR cohort data, and it underwent the same procedure of identifier encryption and pseudo-randomization of the genotype data. And since the results for height were already available because a previous study was already conducted, we could see whether our findings overlapped. And if the procedure was successful, then we would expect an overlap of 1, and that turned out to be 0.99. So we can conclude that the OSSC enabled us to link sensitive data, the genotype data from NTR, to also sensitive data from CBS, namely the healthcare expenditure data, in a way that protected the privacy of the participants. And we did that on this ODISSEI secure platform that then provided the required high performance facilities that were needed to run the large-scale analysis that we wanted to run, and which were not possible in the regular CBS remote access environment.